Hello students, this is Dr. Faitan Mirza and I am explaining the topic of oxidative phosphorylation here. What is oxidative phosphorylation? We know from Krebs cycle, all that's left of a glucose molecule are reduced hydrogen carriers which include 10 reduced NADs and 2 reduced FADs. What happens in oxidative phosphorylation? We know there are two processes combinedly are added together to carry out oxidative phosphorylation. One is electron transport chain and the other is chemiosmosis. Both the process occur together on the intermembrane, in the inner membrane of the mitochondria. These compartments I'm mentioning to you are being shown diagrammatically here. This is the cytoplasm of the cell, this is the outer membrane of the mitochondria, this is the intermembrane space, and this is the inner membrane of the mitochondria. I'm trying to just magnify this region, which I've shaded I've highlighted this in the box here. So this is the phospholipid bilayer, the inner membrane of mitochondria. And this intermembrane space already discussed previously, it's having the high H plus concentration, and this region is the matrix of the mitochondria having low H plus concentration. This is the ATP synthase enzyme which can produce ATP by combining ATP and PI together. And H plus can pass through it by the process of chemiosmosis. These blocks that I've added here are the electron carriers. So a total of 5 complexes are there, which forms the respiratory complex present in the inner membrane of mitochondria where ATP is produced. Reduced NAD and reduced FAD, they both interact with the first and the second respectively. These are electron carriers. These electron carriers, they take hydrogen away from the reduced NAD and oxidize NAD. And the second one will do the same for the reduced FAD. So reduced FAD loses hydrogen to the second electron carrier and hydrogen is removed from it. The hydrogen which are stripped from here makes the NAD and FAD oxidized, and these hydrogens are then split into their component electron and proton. The electrons then start to move along the electron carriers 1, 2, 3, and 4, which are arranged in a series of decreasing energy level. This is at the highest energy level, this is at the lower energy level, this is at a much lower energy, and this is at the lowest energy level of all the 4. This is pretty much similar to how your shells are arranged in an atom and how electrons travel in from one shell to another shell, or probably just subshells or orbitals if you can recall, that they are not all at the same level. So electron, it starts to lose energy and it keeps moving from one electron carrier to the next. And as the electron moves from one electron carrier to the next, it jumps from one carrier to the next and then the next and then the next. Electron keep losing energy. The energy lost by these electrons is used to push the H plus ions into the intermembrane space against the gradient, where already there was a high H plus concentration of H plus ions. And in the matrix, there is a low H plus concentration of. So here, this is the matrix. The electron eventually falls into the matrix and it loses the energy. This was electron transport chain. Inhaled oxygen is present here, so inhaled oxygen ends up combining this electron, and in aerobic respiration, terminal electron acceptor is our inhaled oxygen. 3H plus ion then move from the intermembrane space to the matrix of the mitochondria by chemiosmosis, and ADP and PI combine to form ATP. H plus oxygen and electron combine together to form water molecule. The overall equation for water formation is 2H plus combined with 2 electron and half oxygen molecule to form water. So this is actually what happened. The inner membrane was having respiratory complex, which are 5 membrane proteins, and these include 1, 2, 3, and 4, which are electron carriers, and 5th is the ATP synthase. The 4 electron carriers are arranged in the order of decreasing energy level, and electron carriers are cytochromes. They have RNA and they are glycoprotein in nature. So this is just a text that already I have discussed with you, how electron transport chain occurs. So you need to just describe how electron transport chain occurred, what was the use of electron transport chain, and how the electron was losing energy and how that energy loss was used to push the H plus ions into the intermembrane space. And how this is followed by chemiosmosis, and the eventual outcome of chemiosmosis is ATP being produced by oxidative phosphorylation, and how oxygen serves as a terminal electron acceptor. What is the potential of producing ATPs in oxidative phosphorylation from these reduced hydrogen carriers? One reduced NAD can produce up to 3 ATPs and one reduced FAD can produce up to 2 ATPs. So a total of 10 reduced NADs were there and 2 reduced FAD were there from one glucose molecule. So if you multiply 3 by 10 and 2 by 2, we have a total of 34 ATPs which can be potentially produced during oxidative phosphorylation, because these number of reduced NAD and reduced FAD were produced for one glucose molecule. But energy is used to pull in ADP and PI from cytoplasm back into the matrix so that it can be resynthesized later. And since this energy is used up, so the actual potential of ATP being produced for per reduced NAD and per reduced FAD decreases. So one reduced NAD ends up producing 2.5 ATPs instead of 3, and one reduced FAD ends up producing 1.5 ATP instead of 2 ATPs. So now if you multiply the value of 2.5 by 10 and 1.5 by 2 and add the values together, we get 28 ATPs. So 25 ATPs are produced by reduced NADs and 3 ATPs are produced by reduced FAD. Together, they make up 28 ATPs which are produced in the oxidative phosphorylation. That's it from my side. Thank you so much.